In this procedure, an in vitro culture is set up to study how cell migration from the ganglionic eminence to the cerebral cortex is affected by different fractions of the cortical protein extract. First, latex microspheres are incubated with individual fractions to prepare sources of slowly released proteins. A ferret brain is dissected out and sectioned coronally into thick slices. Tissue explants are punched out from ganglionic eminences. Three-dimensional cultures are set up containing the microspheres and explants embedded in extracellular matrix. After a period of incubation, time-lapse image recording is conducted under a bright field microscope.
The main advantage of this technique over existing methods, like using a chemotactic chamber, is that the movements of individual cells can be observed and quantified.
This method can help answer key questions in the field of development of the nervous system, such as chemotactic and chemokinetic control of motility.
Generally, individuals new to this method will struggle because it is difficult to handle the small tissue explants with the matrigel.
To begin, gradually thaw the frozen matrigel on wet ice to ensure that the temperature remains low enough to prevent premature solidification of the matrix. While the matrigel is thawing, filter sterilize and then slush freeze 500 milliliters of artificial cerebrospinal fluid, or ACSF, by placing a plastic flask in the -80 freezer. When a thin layer of ice forms on the walls, break it off by shaking the flask. Then allow another layer of ice to form, repeating until an ice slurry is created. Next, prepare neurobasal medium according to the manufacturer's instructions and sterilize by filtration through a 0.22 micrometer filter membrane. Incubate at 37 degrees Celsius until ready for use. To adsorb the protein fraction onto the fluorescent latex microspheres, or beads, for diffusion into the matrigel, add 10 microliters of the beads to Eppendorf tubes separately containing 100 to 200 microliters of each of 5 different protein fractions. Incubate in the dark for 30 minutes at room temperature on a shaker. After the 30-minute incubation, spin the beads down using a benchtop centrifuge at maximum speed for 1 hour. After deeply anesthetizing the animal with euthyosol, dissect out the whole brain and submerge it in the previously prepared ice-cold ACSF. Fully separate the hemispheres using a scalpel and slice coronally at 500 micrometers using a tissue chopper or similar device, collecting the slices into a small petri dish containing ice-cold ACSF and sitting on wet ice. Using a dissection microscope, identify slices containing the ganglionic eminences, or GE, and trim away regions of cortex that may interfere with subsequent steps. Press a Harris unicore bore in the area close to the ventricular surface of the GE, retract a core containing predominantly the ventricular zone, and expel it with a plunger into the ice-cold neurobasal medium. Transfer a batch of explants from the medium into a 0.5 milliliter drop of ice-cold matrigel placed in a separate dish, taking care to have the explants fully immersed and avoiding bubbles. Keep the dish on wet ice. Working in a tissue culture hood, prepare culture-grade 6-well plates by dotting each well in its center with 2 microliters of cold matrigel using a 20 microliter pipette tip and placing an autoclaved round 18 mm glass cover slip on each drop. Remove the supernatant and resuspend the beads in 10 microliters of neurobasal medium using a vortexer. Add 30 microliters of the ice-thawed matrigel to the suspension, mix well, and place on ice. With the aid of the dissecting microscope, place 1 to 2 microliters of the bead suspension using a 20 microliter pipette and let it set for 1 to 2 minutes, being sure not to let it dry out. Pick up 2 explants from the matrigel drop and place them in the desired vicinity of the bead deposit 0.5 to 1 mm away, symmetrically and on opposite sides. Let set for no longer than 5 minutes at room temperature. Cover the beads and the explants with a flat layer of about 30 microliters of matrigel and place in a 37 degrees Celsius tissue culture incubator for 15 minutes. After incubation, add 2 milliliters of neurobasal medium and then place back in the incubator for 3 to 4 days, monitoring migratory activity daily. After incubation, choose cover slips of interest with explants that have generated a radially uniform cloud of cells. Place chosen cover slips into a fresh culture plate and incubate overnight. Before imaging, use a sterile spatula in a tissue culture hood to move the cover slip in order to release any microbubbles from under the cover slips. Fill the space between the wells with water to prevent drying and carefully transfer the plate to the microscope. Using imaging software, define an imaging scheme, being sure to take into consideration the 3D nature of the culture by collecting a Z-plane series. Once the parameters are set, initiate time-lapse imaging. Representative time-lapse recording of a ganglionic eminence explant generating migrating cells is shown here. Cells migrate radially out of the tissue. Their attraction to the protein source is variable depending on the nature of the source. In this higher power movie, individual moving cells can be easily traced, as indicated by arrowheads. Many cells are seen branching, a few of these are indicated with arrows. Tracing of individual cells yields positional information that can be further analyzed. Example plots show a differential response between fractions A and D, as evidenced by distinct trajectories of cells leaving the explant.
Once mastered, this technique can be done in 4 hours if it is performed properly.
While attempting this procedure, it's important to remember to keep matrigel on ice.
After watching this video, you should have a good understanding of how to prepare a slow-release source of proteins, set a three-dimensional tissue culture, and conduct time-lapse imaging in order to observe and analyze movements of individual cells.